Here's how tetraploidies go. You begin with a string of genes. You double like the tetraploid. You lose one or the other, but not both. So you can't call it gene loss. If you call it diploidization, that's not too bad if you have a concept of what the diploid is. It's actually more like ancestralization. I call it fractionation. And you end up with a fractionated pair of chromosomes. Some of them, however, some of these genes are pairs. That's a pair. But most of them are fractionated. There's just one left. And then they duplicate again, just like in the Arabidopsis lineage after the branch with papaya. And they go through the same process. And then you use an outgroup, say grape. This is their relationship synthetically. So this is just a little animation to show you the principle of the thing. And here's what it really is. It takes 4 regions of Arabidopsis, on 4 different chromosomes in this case, to cover just one little region of either grape or papaya. So here's a bunch of genes in grape, a bunch of papaya, all those little green things show you the collinearity of this region. However, with Arabidopsis, as you might guess, fractionation has been, there's been 2 rounds of tetraploidian fractionation, sticking this whole thing together, the whole genome is like this. Using our software, this is all homemade software, available to you as Koji. I'll mention that again in a second. You can get a feeling with the symptometer for how syntenic they are. You can find inversions easily with the software. This is all on the fly. When you put things together in our software, you don't deal with things like browsers. It's not static info. You set them up, and then the thing cranks in front of you, and you get a little eye candy, a little double helix rolling around, and then you get the result. If you don't like it, change the parameters. Don't use BLAST, use chaos, use dialine, use whatever you want. Local duplications, you can tell gene losses. Bad annotations are absolutely apparent. You find that the genes in Arabidopsis are well annotated, experimentally supported, and they have an exon showing there. The exon's missing in grape and papaya, but the hit's there. Most likely they missed it. So you can prove it. It's not ab initio annotation, but it still gives you a feeling for just how good or bad the annotation is of any organism if you can compare it to their better annotated relative, and also have excellent visualization of the blast hits. So the genes that are left after tetraploidy are not a random set of those going in. So this all has to do with just that last tetraploid in Arabidopsis, but it's also been repeated in rice and in several other tetraploidies. A number of labs showed this. I was actually a latecomer to the group of people who showed in Arabidopsis that it was a special group of genes. I added a few great groups of genes, like the proteasome cores. But basically, Vanderkeer's group, Ken Wolf's group, Sonia Gering's, and my group all contributed our little bit to the fact that genes that are particularly, the genes that interact at the protein level with other gene products of the same type to make like ribosomes, proteasomes, motors, transcription factor complexes, signal transduction, phosphatases, and protein kinase activities known to be involved in signal transduction. Groups of proteins that are known to have a lot of interactions tend to be in this list of those that are hard to get rid of. It's not that they're selected to be paired. It's that, of course, you try to get rid of everything that has no function, but you can't get rid of it, and the theory is very clear. The theory is that because of the gene balance hypothesis, that's a hypothesis of Birchler, Etienne, and Hurst, and others, You don't get rid of your connected genes because to get rid of them throws the genome into imbalance. It likes to have its parts all in the same stoichiometry, and so if they all play together and you take away one, that's not good. If you could take them all away one dose or add a dose to all of them at once, that would be great, but you can't do that. So therefore you're stuck and you get driven up for your connected genes at every tetraploid. If you use the same logic every tetraploid, you get your connected genes, your most stoichiometrically confined genes, driven up in concentration. The opposite should be of tandem duplications. If you take a regular genome and then you take one gene and duplicate it, that should be okay if it doesn't play with others. If it just is a loner gene, so what? You make all the copies you want, you make all the copies you don't. You could even select for just that gene alone. But if it plays with others, you would also have to duplicate in tandem its playmates. That's hard to do, so you just don't tandemly duplicate those. So if this is all correct and the gene dosage hypothesis is the correct explanation, there should be an inverse relationship between the ability of a gene or gene group to be tandemly duplicated and the ability of the gene or gene group to be duplicated over by tetraploidy, retained by tetraploidy. And that turns out to be the case for transcription factors in Arabidopsis after alpha. Tandem repeat frequencies, not the number of tandems, but the number of tandem events. Down here, it's the frequency of genes retained post the most recent tetraploid. This is probably more convincing. All of the Go categories in Arabidopsis are arranged. If they have 70 categories, have more than 40 genes. They're arranged from the 6% at the bottom, 6% are retained post-tetraploidy. We assumed throughout all that the reason that, say, transcription factors were retained after a tetraploidy was the same as the reason why ribosomal genes, or proteasome genes, or motor genes encoding motor proteins were retained, and that has to do with connectedness of their product. But it doesn't have to be that way. It could be that it's the connectedness of the gene's adjacent sequences, that it's in the number of cis-acting binding sites they have, that the player could be a stoichiometrically important or concentration-dependent component in telling a gene how connected it is and thus how resistant to being removed after tetraploidy. Could easily have the DNA binding site be in the equation. And we do have some evidence for that. And as especially in rice, if you take a gene in rice and compare it to sorghum, you can count its conserved non-coding sequences. And as those sequences go up, so goes the chance that it was retained after, it was retained as a pair. That very gene is part of a pair from the pre-GRAS tetraploid. So you can do it at the gene level, and it gets up very high. That is, here is the average number of CNSs per gene, and then as this little bar, and this is retention, and it does go up, and then as you get to the top 15 or the top 56 or the top 340, your retention goes up to 40%, 45%, 39%. Definitely, conserved non-coding sequences seem to be at least a viable candidate for being part of the connectedness picture. I want to study fractionation mechanism, whatever is removing those genes. And sorghum split with maize about 11 million years ago, and shortly thereafter, like within a million years, maize lineage had a tetraploid. That's largely fractionated. The maize genome came out about a month ago, and little peaks of the genome, unannotated genome, and assembly came out before that, and we've been busy working on that. We figured it should be special, that the tetraploid would somehow make for a special mechanism. That's why That's the hypothesis. There's a whole raft of papers that say, following the tetraploidy, there's lots of breaks, there's confused chromosomes, there's non-additive transcription, non-additive methylation. Everything goes wonky. I figured then that the mechanism that removes genes following the tetraploidy might itself be a special mechanism. We'll see if that's true.